ICRISAT, south of Hyderabad, they're trying to optimise plants, especially the ones for semi-arid tropical regions. And they're optimising plants in various ways, and one way is to introduce genes for drought tolerance into plants that aren't drought tolerant enough, such as groundnut. So how does this process begin?
So this basically is, we call it genetic engineering, which is to complement plant breeding activities. So here we can cross the species barriers, which means we can bring genes from related plants which have got resistance levels, and then introduce them into our crop of our interest. In this case, it's groundnut.
So where would you get the gene for drought resistance if it doesn't already exist in the groundnut? Which plant would you take it from?
So in this case, we have got a gene from Arabidopsis. It's a small crucifer plant which has got drought tolerance level.
And so this is a very well-studied plant, so you already have the molecular marker,
right,
so you know where the gene is.
So we know the gene which is present, so all we're doing is taking a gene from Arabidopsis and putting it in peanut or groundnut, and then looking whether our groundnuts also become tolerant like.
Okay, so let's have a look and see what's going on in the lab.
So the first step in this process is having a good tissue culture system.
Okay, yeah.
Where we can take different plant parts.
Okay.
And then optimize it to get shoot regeneration.
Okay, lovely. So that's what these people.
So here in this case we've got a regeneration system where we can obtain large number of plants in tissue cultures. And this is where you introduce the gene.
So the gene is being introduced, and then you have to tell whether or not the plant that you've created has that drought tolerance gene. And
so this is where she is basically trying to look for plants which have got our gene. And once we've selected the plants on a selection medium, then we go and cultivate the other plants.
so then you cultivate the other plants.